Hello everyone, welcome to 10-Minute Physiology. In today's video, we're going to be talking about the effects of ANP on sodium reabsorption. So with that, let's give it a go. So let's begin with the first segment of the nephron, which is the proximal tubule. So how does ANP affect sodium reabsorption in the proximal tubule? Well, in order to understand how ANP affects sodium reabsorption, we need to look at another signaling molecule first. And the reason why is because in order for ANP to exert its effects on the proximal tubule, we need to have another signaling molecule present. And this signaling molecule is angiotensin II. So angiotensin II can bind to the AT1 receptor. And the AT1 receptor can be coupled to a number of different G proteins. And in this specific case, this AT1 receptor is coupled to a GI protein. So when angiotensin II binds to the AT1 receptor, this activates the GI protein. And the GI protein will modulate the activity of another enzyme. And this enzyme is called adenylate cyclase. So adenylate cyclase is an enzyme that catalyzes the conversion of ATP into cyclic AMP. And when GI interacts with adenylate cyclase, this will decrease the activity of adenylate cyclase and therefore decrease the amount of cyclic AMP. So when we decrease the amount of cyclic AMP, this will decrease the activity of protein kinase A. And when the activity of protein kinase A is decreased, this will have a specific effect. So protein kinase A can phosphorylate specific regulatory proteins. However, when the activity of protein kinase A is decreased, this decreases the amount of phosphorylation that pKa does to the regulatory proteins. So when the amount of phosphorylation of these regulatory proteins is decreased, this will result in an increase in the activity of the sodium-hydrogen exchange protein, therefore allowing the sodium-hydrogen exchange protein to bring in more sodium from the tubule lumen. In addition, these regulatory proteins will also increase the activity of the sodium-potassium pump, which allows the sodium-potassium pump to bring more sodium from the cytosol into the interstitial fluid. So this is going to be one of the effects of angiotensin II. Angiotensin II can increase the amount of sodium reabsorption that takes place in the proximal tubule. So now that we know what angiotensin II can do, let's look at what ANP does. So ANP will bind to the ANP receptor. And when ANP binds to the ANP receptor will convert GTP into cyclic GMP. Cyclic GMP can then increase the activity of another enzyme, specifically protein kinase G. So in order for protein kinase G to affect the regulatory proteins, we need to have angiotensin II present. And the reason why is because in order for ANP to exert its effects, angiotensin II must be present. And this is because angiotensin II can decrease the activity of protein kinase A. And when the activity of protein kinase A is decreased, this keeps protein kinase A from phosphorylating the regulatory proteins. And when protein kinase A is kept from phosphorylating regulatory proteins, this allows protein kinase G to phosphorylate these regulatory proteins and therefore exert its effects on these two transporters. So in other words, in order for ANP to exert its effects, Ang2 must be present because Ang2 keeps protein kinase A from phosphorylating these regulatory proteins. And if protein kinase A is kept from phosphorylating these regulatory proteins, this allows protein kinase G to phosphorylate these regulatory proteins and exert specific effects. So when protein kinase G phosphorylates these specific regulatory proteins, what happens is the regulatory proteins will actually decrease the activity of the sodium-hydrogen exchange protein. So when the activity of the sodium-hydrogen exchange protein is decreased, this means that the sodium-hydrogen exchange protein brings in less sodium. In addition, the regulatory proteins will also decrease the activity of the sodium-potassium pump. Therefore, the sodium-potassium pump will bring out less sodium into the interstitial fluid. So as a result, ANP will decrease the amount of sodium reabsorption that takes place in the proximal tubule. So in summary, ANP and BNP can only exert their effects on sodium reabsorption in the proximal tubule when angiotensin II is present. And this is because in order for PKG to phosphorylate the regulatory proteins, the activity of protein kinase A must be decreased. Now, angiotensin II decreased cyclic AMP, which decreases the activity of protein kinase A. This decrease in the activity of protein kinase A increased the activity of NHE3 and the activity of the sodium-potassium pump. However, ANP has the opposite effect. ANP increases the activity of protein kinase G, and through the same regulatory proteins, it decreased the activity of the sodium-hydrogen exchange protein and the sodium-potassium pump. So the next section of the nephron that we're going to look at is going to be the thick ascending limb of Henle. So how does ANP affect sodium reabsorption in the thick ascending limb of Henle? Well, when ANP binds to the ANP receptor, the ANP receptor will convert GTP into cyclic GMP. Cyclic GMP will then activate another protein called phosphodiesterase 2. And phosphodiesterase 2 hydrolyzes cyclic AMP into AMP. And as a result of this, the amount of cyclic AMP inside the cell is decreased. This decrease in the level of cyclic AMP will decrease the activity of protein kinase A. And when the activity of protein kinase A is decreased, this will decrease the activity of NKCC2. So if the activity of NKCC2 is decreased, this means that NKCC2 brings in less sodium into the cell. So therefore, it decreases sodium reabsorption. In addition, the decrease in the activation of pKa will result in a decrease in the implantation of NKCC2 proteins in the apical membrane. So in other words, ANP decreases the activity of NKCC2 and decreases the amount of NKCC2s in the apical membrane. Together, these effects will result in a decrease in sodium reabsorption in the thick ascending limb of Henle. So ANP or BNP decreases the activity of NKCC2 and the number of NKCC2 transporters in the apical membrane. And as a result, ANP decreases sodium reabsorption in the thick ascending limb of Henle. So the next segment of the nephron that we're going to look at is the cortical collecting duct. So how does ANP affect sodium reabsorption in the cortical collecting duct? Well, when ANP binds to the ANP receptor, this causes the conversion of GTP into cyclic GMP. Cyclic GMP will then activate protein kinase G, and then protein kinase G will basically inhibit the ENaC channels. So when the ENaC channels are inhibited, this means that less sodium will be brought into the cytosol, and therefore sodium reabsorption is decreased. So in summary, ANP and BNP inhibits ENaC channels in the cortical collecting ducts, which decreases sodium reabsorption in the cortical collecting ducts. So the last segment of the nephron that we're going to look at is the inner medullary collecting duct. So how does ANP affect sodium reabsorption in this region of the nephron? Well, when ANP binds to the ANP receptor, this results in GTP being converted into cyclic GMP. Cyclic GMP will then activate protein kinase G, and then protein kinase G will inhibit the CGGC channel, which means less sodium will be brought into the cytosol from the tubule lumen. In addition, cyclic GMP also acts as an allosteric modulator on the CGGC channel. And as a result, when cyclic GMP interacts with the CGGC channel, this means that the CGGC channel will bring in less sodium and therefore decrease sodium reabsorption. So ANP and BNP inhibit CGGC channels through allosteric modulation via cyclic GMP and through the action of protein kinase G. Therefore, ANP decreases the amount of sodium reabsorption that takes place in the inner medullary collecting duct. So that's it for this video. I hope this video helped you understand the effects of ANP, and I hope to see you in the next video. Thank you for watching, and I hope to see you next time.